thought to make a short video to explain the complement system. We have three pathways. The first one is called the classical pathway, and the second one is called the lectin pathway, and the third one is the alternative pathway. The first classical pathway, you should know that this happens in the case of a secondary infection, because it starts with the antibodies. So since there are antibodies, it means it's a secondary infection. The lectin pathway and the alternative pathway both can happen in the primo infection. Okay, and the primo infection is possible. So let's start with the classical pathway and see how it happens. So when the antibody is bound to the antigen on the surface of the pathogen, what it does, another protein can come. It's called C1q. C1q will go ahead and recruit two other proteins on the surface. They are C1r and the C1s. These two proteins have an activity to do what? To take the C2 protein and the C4 protein and change it into C2. Maybe make it bigger here. They can make it into C2a and C4a. Okay, and there, this is the small, this is one of the subunits, and this is the second subunit, the B, C4b. And in this case, I'm gonna do like a comparison here. In this case, it is not this, in the lectin pathway, it is not the C1r and the C1s who do this. It is a different mechanism. Instead of the antibody, there is a protein called mannose binding protein, which will recognize the mannose sugar on the pathogen, and it will recruit two proteins which work similarly to what these SC1R and C1s did. These are mannose associated protease, mannose associated protease something, I forgot the name, let me check. Yeah, MAPS. Serine proteases, okay? Serine protease. So these, what they do the exact same thing. So if a C2 and the C4, they both come here and they get cleaved and they become like this. On the other hand, in the alternative pathway, we will not have these two, which neither an antibody or a mannose binding protein. What will happen instead is the C3 will spontaneously cleave into C3a and the C3b. C3b will come and sit on the pathogen, and when it does that, it recruits two other factors. We have a factor called factor B and another factor called factor D. What will happen here is this factor B will cleave, so the B will become BA and BB, as we will see in how they are cleaved into two types, two subunits. So now what we have, in this case, we have the C2 and the C4 cleaved. Here, in this case, we have the CB cleaved. So let's continue what happens here. In this case, it's a very similar scenario what is happening. This subunit here and this subunit here will come and make a complex called C4b2a, okay? C4b2a. On another name, we can call it C3 convertase. What C3 convertase does, it takes C3 and converts it into C3a and C3b. Okay, so in here, what is happening? How are we gaining this C3 convertase? So here we have the C3b that already exists, and we have the big subunit right here, the BB, making this complex, which is the C3 convertase of the alternative pathway. Okay, so we have two types now of C3 convertase. It's one here and one here. After the C3 convertase, we need the C5 convertase. How do we get that? So what will happen in the first scenario here, this subunit, the B subunit again, will come and join this complex. So we will have now C4B2A3B from this. This is named also C5 convertase. What happens here? Here, this is a C3 convertase, so it will take C3 and will give us C3A, C3B, and this 3B will come and join this complex, C3B, BB, 3B. It's a bit of a weird name, but yeah, here you go. This is a C5 convertase now. Okay, now we have C5 convertase on this side, and we have a C5 convertase on this side. So what C5 convertase does is takes the C5, and so in both ways, now the C5 will become C5A and a C5B. The C5B will come on the pathogen. Okay, C5B will recruit other proteins. It will recruit C6, C7, plus C8. All of these will help the C9 to make 14 to polymerize into 14 subunits, and they will make a pore into the pathogen. This is called all of this is called the membrane attack complex, the MAC or CAM in French. Okay, so this is the how these pathways are all leading to the lytic pathway. So the lysis will happen. Okay, after that this MAC was formed. Now there is a little detail before I go further. There is a little detail. So we always talked about how this subunit, how this B subunit goes and joins the complexes. What happens to the little C2B and all the other A subunits? So only this is an exception, whereas the B, but all of the other A subunits come. So we have the C2B, C3A, C5A, and you have the other A the C4A as well. So all of these subunits, we call them anaphylatoxins. What they do a couple of stuff. They do a couple of stuff. They work on the endothelial muscles of the blood vessels and make them more permeable, so the cells can go through them. In the end, they activate cytokines and chemokines, and they attract other cells. What all this does is the activation of inflammation. Okay, so you should know these subunits will cause inflammation, while these subunits will call the membrane attack complex and leading to the lysis of the cell. Good luck.